Welcome back to the online class. Our topic is development of embryo sac, and this process is known as megagametogenesis. In the last video, I have explained you about the process of megasporogenesis, how we get 4 megaspores from one megaspore mother cell. Yes, so out of these 4 megaspores, only one of the megaspore is functional and the other 3 they degenerate. In majority of the flowering plants, only one of the megaspores is functional, while the other 3 degenerate. Here, degenerate means they will disappear. Only the functional megaspore develops into female gametophyte, which is also known as embryo sac. And this method of embryo sac formation from one single megaspore is termed as monosporic development. As here only one single megaspore is involved, so it is known as monosporic development. So remember this term, monosporic development. So now let's see how embryo sac develops from one megaspore. Suppose if this is the functional megaspore, we will discuss how this functional megaspore will develop into an embryo sac. So we shall discuss this process step by step. First of all, the nucleus of the functional megaspore will divide mitotically to form two nuclei, which move to the opposite poles, forming the two nucleate embryo sac. So as you can see in this first diagram that there are two nucleus, so it is known as two nucleate states. So both the nucleus are at the opposite poles, 1, 2. Two more sequential mitotic nuclear divisions result in the formation of 4 nucleate and later the 8 nucleate stages of embryo sac. So after these two nucleate stages, these two nucleus again it will undergo mitosis type of cell division, and each of them they will give two daughter nucleus, and so 2 plus 2 we will get 4 nucleus. So it is called 4 nucleate stage. Again, after the 4 nucleate states, this nucleus they will again undergo mitosis type of cell division, and we will get 8 nucleus, 2 from each of them. So total it is 8, and therefore it is known as 8 nucleate stage. Up to here we can see in the diagram that all these nuclei are arranged in the opposite poles. 4 of them are on the upper side and the other 4 are on the lower side, so they are in the opposite poles. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the central cell. So now look at the diagram. Here the two opposite poles are the micropylar end and the chalazal end. Okay, so out of the 8 nuclei, 6 of them they will be organized into cells. 3 nuclei will be on the micropylar end and the other 3 will be on the chalazal end. And the remaining 2 nuclei, which is known as polar nuclei, will now come to the central cell, which is Lars. As it is the middle part of the cell, so it is known as Lars central cell. Now let's see this diagrammatic representation of the embryo sac. As we already came to know that 6 nuclei are organized into cells, and out of 6, 3 cells are grouped together at the micropylar end and constitute the egg apparatus. So this egg apparatus, it consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus. Okay, there is a filiform apparatus in the synergids, and this filiform apparatus they help in guiding the pollen tubes into the synergid. Now let's look at the diagram. So first of all, see the micropylar end. Here we can see there are 2 synergids, 1 and 2. Okay, and also there is a egg, and in the synergids there is a filiform apparatus which will help the pollen tube to enter the egg. And the other 3 cells are at the chalazal end and are called the antipodals. And the large central cell has 2 polar nuclei. Now come back to the diagram and look at the chalosal end. Here we can see there are 3 cells, and these 3 cells, they are together known as antipodals, and they are at the chalosal end. And in the central cell we have the 2 nuclei, and these 2 nuclei, they are organized into a single cell. Both of the nuclei will be inside one single cell, which is known as polar cell. And this polar nuclei is situated just below the egg cell. Now let's count the number of nucleus and number of cells from this diagram. Okay, so first of all, let's count the number of nucleus. 1, 2, 3. So 3, 4, 5, 6, 7, 8. So we have total 8 nucleus. And now let's count the number of cells. Cells is how many? 1, 2, 3, 4, 5, 6, 7. So we have 7 number of cells. Thus, at maturity, a typical angiosperm embryo sac is 7-celled and 8-nucleate. So therefore, it is known as 7-celled 8-nucleate stage. So with this, we have completed the topic pre-fertilization structure and events, where we have discussed about the male gametophyte and female gametophyte in details.